Welcome to this explainer. We're going to jump right in today and unpack PlacentaCellEnrich. This is an absolutely incredible web-based tool put together by the Tutor Lab, specifically designed to characterize placenta cell-specific gene enrichment. If you're out there working in genomics or reproductive biology, you already know how absolutely crucial it is to figure out exactly where and how genes are expressing during pregnancy. Well, today, we're going to show you exactly how this tool can streamline that entire workflow for you. But first, let's talk about the elephant in the room. How on earth do we actually characterize these massive placenta gene sets? If you're a researcher analyzing pregnancy data, you know the drill. You're constantly left staring at a spreadsheet with thousands of genes. Trying to map that massive, complex list to very specific cells in the placenta and decidua, it's like finding a needle in a haystack. You have all this data, sure, but making real biological sense of it, pinpointing exactly which cells are driving the phenotypes you're observing, is a massive bottleneck. Enter PlacentaCellEnrich. This is basically your bioinformatics shortcut. It was custom built to provide a highly reliable, efficient, and honestly super user-friendly interface. It completely empowers you to take that intimidating wall of data and instantly interpret your complex gene sets. And the best part? You don't need to be a computational wizard writing your own custom scripts from scratch to do it. Okay, so here's our game plan for this explainer. First, we'll look at the foundational datasets. Then, we'll break down how we define cell-specific genes. Next, we'll actually take the tool for a spin. And finally, we'll talk about interpreting your results. All right, let's dive right into Section 1, the foundational datasets. You know, any enrichment tool is really only as good as its background data. PlacentaCellEnrich is powered by 3 distinct major placental single-cell RNA-Seq studies that act as your reference map. First up, we've got the Suryawanshi study, which features 22 specific placental and decidual cell types. Then, there's the Vento-Tormo dataset covering 32 cell types. And finally, the Arutyunyan dataset, which brings a staggering 42 distinct cell types to the table. Having these 3 separate, robust datasets gives you incredible flexibility and real confidence when you're analyzing your own lists. Moving right along to Section 2, defining cell-specific genes. So how does the math actually work here? How does the algorithm classify a gene as belonging to a specific cell? Well, taking a cue from the Human Protein Atlas, the absolute strictest classification they use is called Cell Enriched. For a gene to earn this label, it literally needs a massive 5-fold expression spike in one single cell type compared to all other cells in the dataset. If a gene is cell-enriched, it's basically a glowing neon sign pointing directly to that specific cell type. But, you know, biology is really that isolated, right? Sometimes genes like to hang out and be highly active in a small family of related cells. So if that high expression, and again, we're talking at least a 5-fold higher level, is shared across a tiny biological neighborhood of 2 to 7 cells compared to everything else, it gets labeled as group enriched. It's still highly specific, it's just specific to a specialized cluster rather than one lone cell. And finally, we've got the cell-enhanced classification. This identifies genes with an expression level 5 times higher than the overall average of all other cells. So they might not quite meet the strict absolute criteria of the first 2 groups. You know, maybe there's a little bit of noise or lower level expression happening elsewhere, but they are absolutely showing a massive, biologically relevant enhancement in a particular cell type. To sum all of that up, when PlacentaCellEnrich calculates your data and tells you it found cell-specific genes, it is specifically bundling together these 3 heavy hitters. It filters out the background noise, ignores the universally expressed genes, and hones right in on the cell-enriched, group-enriched, and cell-enhanced categories to deliver your results. Let's move on to Section 3, using the tool. Okay, so how do we actually run our data through this engine? The interface is beautifully straightforward. Step 1, just feed it your gene list. You can paste it in 1 per line or easily upload a text or CSV file. Step 2, lock in your parameters. That means picking your organism, the type of gene ID you're using, and which of those 3 reference datasets you want to compare your list against. Step 3 gives you a neat little option to add a custom background list if you really want to fine-tune your statistical significance. And finally, just hit submit and let the algorithm do the heavy lifting for you. And boom, within moments, you aren't just looking at some dry, boring text file. The tool generates fully interactive bar charts. That means you can hover right over the bars on your screen to see the exact enrichment scores, the fold changes, and the cell types. Even better, if you click directly on a bar, the tool instantly generates an expression heat map for those specific genes. This lets you visually verify the enrichment across the different cell clusters on the fly. It is incredibly intuitive. Now, what if you're in the lab working with a model organism like mice? No problem. The tool totally has you covered. You simply input your mouse genes, select mouse from the drop-down menu, and pick one of the human data sets. Behind the scenes, the tool automatically translates your input by mapping one-to-one orthologs against the human datasets using the Ensembl version 100 database. It's an amazing feature that seamlessly bridges the gap between animal models and human placental biology. Which brings us to our final section, interpreting your results. Running the data is the easy part, right? Figuring out what it actually means is where the real science happens. To ensure absolute scientific rigor, 0.01 is the strictly recommended threshold value for your adjusted p-values when you're selecting enriched cells. If your adjusted p-value dips below this number, you can be highly confident in the statistical significance of what you're looking at. And you want to pair that p-value with a minimum fold change value of 2. Seeing a fold change of at least 2 is going to guarantee that you are looking at genuine biological variations, and not just some minor transcriptional noise. So, combining a p-value of less than 0.01 with a fold change of 2 or more, that's your golden ticket for reliable data. Here's a quick pro tip for troubleshooting. If you ever run your list and notice that several of your input genes are missing from the output, or the tool says they couldn't be found, double-check your list. Keep in mind that this tool only analyzes protein-coding genes. So if you accidentally slipped some non-coding inputs into your list, the tool automatically ignores them because they simply aren't present in the foundational reference datasets. Oh, and another thing you might spot in your downloaded results is an NA for cell counts. Don't panic if you see this. It only happens if you chose the Suryawanshi dataset for your analysis. It's just because the original authors of that specific study didn't provide cluster sizes in their dataset. The enrichment math itself still works perfectly, you just won't get a raw cell count in that particular column. Which brings us to an absolutely crucial piece of advice, straight from the tool's creators. They highly recommend this as a best practice for any bioinformatics researcher. Don't just run your data against a single dataset and call it a day. Cross-reference your findings across the Suryawanshi, the Vento-Tormo, and the Arutyunyan datasets. Finding consistency across all three sources is quite literally the best way to firmly validate your hypothesis. PlacentaCellEnrich really just shatters the bottleneck between raw, complex RNA-Seq data and actual, actionable biological insights. Armed with a tool this powerful, what fascinating hidden patterns are you going to uncover in your next gene set? Thanks for hanging out with us for this explainer, and happy researching.